So, on to the last part of respiration, well, aerobic respiration anyway, is the electron transport chain. So, remember, this involves different reactions taking place in the cytoplasm and the mitochondria. So, just before we go any further, let's have a little reminder of what's been going on so far. So, glycolysis took place in the cell cytoplasm. The pyruvate then enters the mitochondria, where the link reaction and Krebs take place in the matrix. And the purpose of this video is to look at the electron transport chain taking place on the inner surface of the crista. So, the link reaction and Krebs take place in the matrix, and then these here, the inner membranes of the mitochondria, these folds are called cristae, and that's where the electron transport is going to take place. And then, just to show you this, which is a bit more detail. So if we start up here, we get the glucose being broken down into pyruvate. The pyruvate then enters the matrix where the link reaction and Krebs cycle take place. What happens is carbon dioxide is released, and we also get ATP produced by substrate level phosphorylation. Now, in glycolysis, the link reaction and Krebs, we get NAD reduced, and we also get FAD. Now, remember we said their job was to pick up hydrogens and carry them to the electron transport chain. Well, they're going to carry them here to the inner mitochondrial membranes, known as cristae, because that's the site of the electron transport chain. Now, what also happens in this stage is oxygen comes in and picks up the hydrogen atoms at the end of the electron transport chain, and it produces water at the very end as a byproduct of respiration. As well as that, we can see here that ATP is produced by oxidative phosphorylation in this process. So there is quite a lot going on, but that's where we are at the minute. So getting back to our diagram that we've looked at, we've been through glycolysis, we've produced pyruvate, we've looked at the link reaction, we've looked at Krebs. Now what we need to have a look at is what's happened to all these hydrogen atoms that we've been losing along the way. Because we lost hydrogen atoms up here and the NAD picked them up, we also lost hydrogen atoms in the link reaction and NAD picked them up, and we also lost hydrogens here where the NAD and the FAD picked them up. So let's see what's actually happening and how the electron transport chain comes into this. So here's what we have so far. We've got 10 reduced NAD from glycolysis, Krebs and links, and we've also got 2 reduced FAD from Krebs. We've also made 2 ATP in glycolysis and 2 ATP in Krebs. Now just before we go any further, this is what we've made per glucose. Now, what we still need to do is to produce more ATP, because so far we've only made 4 molecules of ATP, which isn't very much. We also need to get rid of those hydrogen atoms because they are a waste product, and if they build up, it's toxic to the cell. And we still haven't used up oxygen at all. And we always say how much respiration relies on oxygen. So this is what we need to do. We need to find out how we get more ATP, how we get rid of those hydrogen atoms, what actually happens to NAD and FAD, and how to use oxygen in respiration. So in the electron transport chain, these are the main steps. Now there's a diagram to go along with it, and we'll look at both. But let's look at what's actually happening first of all. So firstly, the hydrogen atoms from glycolysis and Krebs are carried by NAD and FAD to the electron transport chain. Now you'll remember here that this NAD and this FAD, those were hydrogen carriers. So when the molecules lose hydrogen, the FAD and FAD pick them up and become reduced, and they transport the hydrogen atoms to the electron transport chain. So you can sort of imagine them as being like a little boat, and the hydrogen atoms are the little passengers on the boat. So NAD and FAD are the boats, hydrogen is a little passenger. Now, they take the hydrogen and they pass the hydrogen along a series of carriers on the surface of the cristae in redox reactions. each carrier is at successively lower energy levels. So as hydrogen and then the electrons goes along the chain, energy is released and it's used to make ATP. Now, there is part of the electron transport chain where hydrogen actually splits up. So you'll remember from year 10 chemistry that hydrogen on the periodic table appears like this. Now, in terms of its number of protons, electrons and neutrons, this is what it's got. It has one proton because it's that number. It also has one electron because they're the same, but the number of neutrons is 1 minus 1, which equals 0. So hydrogen atoms are made up of one proton and one electron. Now that's going to be important because in the electron transport chain, the hydrogen atoms get passed along the carriers, and initially it's the hydrogen atoms, but then they're going to split up, and then at this point the electrons get passed along and the protons then get passed over the top, and eventually they rejoin and the hydrogen gets picked up by the oxygen and we form water. So this bit was basically just to explain that a hydrogen atom is composed of protons and electrons. So because it's being passed along these carriers, which are at lower energy levels, along the way some energy gets released to produce ATP, which was the whole point of respiration. Now, initially the hydrogen atoms get passed along, but at this point the hydrogen atoms split up into protons and electrons and then rejoin later. The final hydrogen acceptor is oxygen. It joins with oxygen to form water. It is catalyzed by cytochrome oxidase. Now, when we're saying it, we mean hydrogen joins with oxygen to form water. So basically at the end, very end of respiration, we have two hydrogen atoms joining up with oxygen to produce H2O. Now sometimes we just put half O2. So this is the final equation that takes place, and that is catalyzed by the enzyme cytochrome oxidase. Now a few other wee notes is that at this stage in glycolysis and Krebs and even the link, where the molecules release hydrogen, the hydrogen gets picked up by the carriers and the carriers become reduced. So what you'll see sometimes is the NAD either being referred to as reduced NAD to show that it has hydrogen, or just simply NADH. Once they release their hydrogen, they become reoxidized. Now, as well as that, we're saying here that as the hydrogen is passed along the chain, energy is released and it's used to make ATP. Each NAD molecule that we have reduced leads to 3 ATP molecules being produced, and each FAD molecule we have leads to the production of 2 ATPs. And finally then, water is a waste product of respiration. Now just looking at this point, each NAD leads to 3 ATP molecules being produced and each FAD leads to 2 ATP molecules. If we go back to this link reaction, that means that this hydrogen, which is given off from this pyruvate, when it's picked up by the NAD and forms reduced NAD, it later leads to the production of 3 ATP molecules. In the Krebs cycle, whenever this reduced NAD goes to the electron transport chain, it makes 3 ATPs. This one makes 3 ATPs. This one also makes 3 ATPs. But this FAD produces 2 ATPs. So actually, as you can see, one of the things that we needed to do here was to produce more ATP. So because we've made so many NADs, or reduced so many NADs, they all lead to the production of 3 ATPs each. So at the end of this, we actually have quite a lot of ATP produced, which we'll try and work out exactly how many molecules we've made. So the electron transport chain, let's have a look at the wee diagram and we'll try and link these little points to the diagram. So this shows the hydrogen atoms being released, and the oxidized form of NAD picks it up and becomes reduced. Now this NAD will then release its hydrogen and become reoxidized, but it will pass its hydrogen along to one of the first hydrogen carriers, which is FP. Now this FP stands for flavoprotein. Now, the flavoprotein picks up the hydrogen and becomes reduced. Next, it releases its hydrogen again and passes it on to the next carrier, which is coenzyme Q. The coenzyme Q picks up the hydrogen and becomes reduced, and then it releases its hydrogen. Only at this point, this is where the hydrogen splits. Now, it then gets passed along a series of cytochromes, which are basically just more proteins embedded within the membrane of the cristae. They pick up the electrons and become reduced, and then again they pass the electrons on, and at this point we've got hydrogen. But to do that, what had to happen was, after the hydrogen splits, the hydrogen protons basically get passed along here, so they bypass this process, and the electrons join back up with the protons to form hydrogen again. Then, this is at the very end, the oxygen, which is the final hydrogen acceptor, comes along, picks up the hydrogen and forms water. So that's the process. Now, you might be wondering a few things. Firstly, what are these? The FP, the coenzyme Q, and the cytochromes. They are hydrogen carriers, and they are found here. So they're embedded on the inner mitochondrial membrane. So you basically have a fold like that, and the NAD transports the hydrogen, it passes it to flavoprotein, then it passes it to coenzyme Q, and then it passes it to the cytochromes, all along there. So the hydrogen basically gets passed along. Now the next thing we might be wondering is why this is slanted. So this is slanted because this is basically their relative energy levels. Now, those of you who have done chemistry will know that sometimes electrons can either get boosted or reduced to different energy levels. And depending on which way they're moving, they can either gain or lose energy. In this case, the hydrogen atoms can do something similar. As they get passed along from the NAD to the FP to the coenzyme Q to the cytochromes, each one's at a lower energy level. So at each stage, when it gets passed along, energy gets released. Now that energy can be used to make ATP at 3 positions. Now, it's only 3 positions if the hydrogen has been carried there by the NAD. So the NADs come along with its hydrogens and pass them along, and that has led to enough energy being released so that 1, 2, 3 molecules of ATP can be synthesized from each NAD. Now, it's slightly different. If we've got FAD, which we only get produced in the Krebs cycle, the FAD will release its hydrogen into this point. So you can see here that after this point, there's only enough energy to make 1, 2 ATPs. Now, in both cases, so whether it's NAD or FAD, bringing the hydrogen to the electron transport chain, the ATP is produced by oxidative phosphorylation. This basically means that ATP is produced by phosphorylating the ADP, and the energy comes from a series of oxidization reactions. So the energy which is released as the hydrogen is passed along in a series of reduction and oxidation reactions leads to the phosphorylation of ATP and produces a molecule of ATP. And that's the electron transport chain. Now, a couple of other points. So remember, the hydrogen atoms can't be allowed to build up in the cells. So they're passed along the carriers, and as they do so, they release energy. And that's because the carriers are at successively lower energy levels. So when we said that here, that's a really important keyword or key phrase that you need to use. And then because they're passing the hydrogen to successfully lower energy levels, the energy they release can be used to make ATP by oxidative phosphorylation. And then the hydrogen at the very end joins up with oxygen, forming water. And water is the byproduct of respiration. Now, the main products of respiration are carbon dioxide, water, and ATP. Now, the carbon dioxide is breathed out. So your cells produce the carbon dioxide, it then diffuses into the blood. The blood goes to the lungs, and then from the lungs it'll diffuse into the alveoli, and then you breathe it out. The water can either be breathed out, or it can leave the body in urine or sweat. The ATP can then be used for cellular processes like the ones that we talked in the first video, which was all of these processes. Now, just to revisit this point about the hydrogen being passed along, imagine we've got a motorway here, and along the motorway there are 3 little toll bridges. To go through each toll bridge you have to pay, and guess what the cost is? It is 1 ATP. 1 ATP. 1 ATP. So little NAD starts its journey from here, and it travels along this motorway. As it goes along, it passes 3 toll bridges, so it has to pay 1, 2, 3 ATPs altogether. Whereas FAD is a bit sneaky. It's decided to avoid the first toll bridge and decided to just come in here. So because it's come in here, the FAD enters there, and it only has to pay 1, 2 ATPs. So altogether, each NAD will lead to the production of 3 ATP molecules, and each FAD will lead to the production of 2 ATP molecules. Now, this is a little challenge if you haven't done respiration before. So if each reduced NAD produces 3 ATP, each reduced FAD produces 2 ATP, and there's some ATP produced in glycolysis and some ATP produced in Krebs, how many ATPs are produced in total from one molecule of glucose? Now remember, we start with one molecule of glucose. It splits up into 2 pyruvates, and then each pyruvate has to go into the link, each pyruvate has to go into Krebs, and then all the NADs and the FADs from along the way will have to go into the electron transport chain. So see if you can work it out. Now, you could probably use this table to help you out. So let's see if we can work it out. So, location of each process. Cytoplasm, matrix of mitochondria, and then the final one, electron transport chain. You can either say inner mitochondrial membrane, or you can say surface of cristae. Now, in glycolysis, we made 2 reduced NAD. There was no reduced FAD there. In the link reaction, we made 1 NAD, but remember, there was 2 pyruvates, so altogether we made 2 NADs. So we needed to put a cheeky little 2 there. In the Krebs cycle, we made 1, 2, 3 reduced NADs, but remember we need to multiply it by 2, so we made 6 reduced NADs. So we put a 6 there. We also made 1 reduced FAD, but because there's 2 pyruvates, we've got 2 FAD. Now, in the electron transport chain, that's where the FAD and the NAD go to. So we don't actually produce any more reduced NADs or reduced FADs there. Now, after that, and this is what we're after, we want to work out how much ATP was produced. So it can either be produced by substrate level phosphorylation, which was produced as a result of chemical reactions. That only takes place in glycolysis and Krebs. Whereas ATP produced by oxidative phosphorylation, which was basically produced as a result of redox reactions in an electron transport chain. So that only takes place there. Now, in glycolysis, we made 2, and that was net ATP. Now, you'll remember actually we made 4 at the end of glycolysis, but we actually used 2 up at the start. So if I show you that, there's the 1 that we used up at the start, the second one we used up, but then at the very end we were able to produce 2 times 2, which was 4 ATP. So we've got a net gain of 2 ATP. Then in the Krebs cycle, we were able to make 1 per pyruvate, so therefore we made 2 overall. So altogether, we made 4 ATP by substrate level phosphorylation. The next thing we've got to do is work out how many ATPs we were able to make from all these NADs and FADs in the electron transport chain. So altogether, we have got 10 NADH, or 10 reduced NAD, which comes from adding those numbers up. And remember, each NAD produces 3 ATP. So 10 times 3 equals 30 ATP. And as well as that, we also have the 2 reduced FAD. Each FAD will produce 2 ATP, so it's 4 ATP. And just to highlight that and keep track of it, that's what we've got there. So altogether, we have got 34 from the electron transport chain, which was basically because of the NAD and the FAD, and 4 from substrate level phosphorylation. So each glucose molecule produces 38 molecules of ATP. And this diagram just summarizes all that. So we start with the glucose in glycolysis. Remember, that's taking place in the cytoplasm. We get reduced NAD, 2 of them to be precise, and we also get 2 ATP produced, and we're going to put net in brackets. We end up with 2 pyruvate molecules, and from these 2 pyruvate molecules we get 2 carbon dioxide molecules released, 1 from each, and we also get 2 reduced NADs, again 1 from each pyruvate molecule. We then get 2 acetyl coenzyme A molecules, and it gets fed into the Krebs cycle. It gets decarboxylated along the way, and 2 decarboxylations take place. So altogether we've got 4 carbon dioxides. We also get 2 ATP produced by substrate level phosphorylation, and we also get 6 molecules of reduced NAD and 2 molecules of reduced FAD. So altogether we have got 6 molecules of reduced NAD, 2 from glycolysis, 2 from link, and 6 from there. So altogether there was 10 NADH. And we also have 2 molecules of FAD. And along the way, we've also got a few other things. The NAD and the FAD get fed into the electron transport chain, where they get passed along a series of carriers on the mitochondrial inner membrane, or the surface of the cristae. Because the carriers are at progressively lower energy levels, it releases ATP. And to be precise here, because we've made the 2 ATP over here and the 2 ATP over here, there are 34 ATPs produced in the electron transport chain. So the 4 ATP from substrate level phosphorylation plus the 38 ATP from oxidative phosphorylation, 34 ATP from oxidative phosphorylation equals 38 ATP per glucose. Now, as well as that, we've also got oxygen here. Now, at the end of this, we take 6 oxygen molecules, they react with the hydrogen atoms, and we get 6 molecules of water. So, to tie all this up, we've taken glucose, which was C6H12O6. We've added 6 oxygen molecules, and we've got 6 carbon dioxides plus 6 molecules of water plus 38 molecules of ATP. And that is the end of aerobic respiration. That was tough.